Hi everyone, for this experiment, we're doing a titration to find the acid concentration in a commercial soda. The process involves two main parts. First, standardizing our sodium hydroxide solution using KHV, and then using that accurate base concentration to measure the citric acid in our soda sample. For our safety, we already have our goggles and gloves on, because both the acids and the sodium hydroxide used in today's experiment are corrosive. If there's any contact, we'll flush the area with water immediately. We start with exercise 1, standardizing the sodium hydroxide solution. This makes our sodium hydroxide a standard solution with a precisely known concentration. We use potassium hydrogen thalate, or KHB, which is a primary standard, meaning its purity is known. First, we accurately weigh a sample of solid KHB, recording the mass to the nearest milligram in our notebook, which was 0.00458 grams. We're going to be doing at least 3 trials to get accurate and precise results. Next, we dissolve the KHB in about 50 milliliters of distilled water. KHB can dissolve slowly, so we gently swirl the Erlenmeyer flask until the KHB has completely been dissolved. Now for the burette, we prime it by rinsing its insides with a small amount of distilled water first, and then sodium hydroxide solution, and then discarding the rinse. Then we fill the burette completely and record our initial volume reading to be 1.02 milliliters. To see the end point, we add a couple of drops of phenolphthalein indicator that remains clear until the base, sodium hydroxide, is added to the KHB solution. We slowly add the sodium hydroxide from the burette to the KHB solution, swirling as we go. As we get close to the point of equivalence, which is when the solution begins to turn light pink, we slow down the addition to dropwise. We stop the titration immediately when the light pink color appears and stays for about 30 seconds of swirling the flask. We then record our final burette volume to be 19.29 milliliters and repeat this process for 2 other KHB samples to get consistent and precise results. This is exercise 2, determining the acid content in a soda. Since the soda we're using is clear, we know the primary acid is citric acid. Our TAs already took care of decarbonating the soda by heating it and adding the same volume of water that was evaporated. Next, we accurately measure 10 milliliter portion of the pre-treated soda and transfer it to a clean Erlenmeyer flask. We add about 20 milliliters of distilled water. This just gives us enough volume to observe the color change clearly. We finish the preparation by adding 2 to 3 drops of phenolphthalein indicator to each of the 3 trials. Now, we titrate the soda using the standardized sodium hydroxide solution from the burette. Citric acid is a triprotic acid, meaning it released 3 protons, so this reaction is a 3-to-1 stoichiometric reaction with the sodium hydroxide. We titrate dropwise, swirling continuously until we see the faint pink color that signals the endpoint. We record our final burette reading. The technique we used today was titrations, which is a laboratory procedure for quantitative analysis, which is used to find the concentration of an unknown solution. It involves a titrant of known concentration and an analyte of unknown concentration. Before doing your titrations, you need to know the concentration of your titrant, the volume of your analyte, and the balanced chemical reaction. The data you will collect is the volume of the titrant used, and then you will calculate the concentration of the analyte. To read the volume in the burette, position eyes at the bottom of the meniscus and estimate to the nearest 100th place.